Good morning, good afternoon, and good evening, everyone around the world. I'm honored to welcome you all to this webinar interview with the CEO of Anaveon, Dr. Andres Cantobodis. I'm Echo Hindoyan, the CEO at MSQ Ventures, a New York-based cross-border advisory firm specialized in the life science. Dr. Cantobodis has extensive experience in drug development for immune-mediated diseases, having taken several modalities to clinical trial and later stage development. He founded Anaveon in 2018, bringing with him a wealth of expertise in drug development and commercialization. In this webinar, we will be discussing a range of topics related to Anaveon's work, including the company's approach to drug development, its progress in clinical trials, and its plans for the future. We will also explore the challenges facing the pharmaceutical company today and how Anaveon is positioning itself to address these challenges. We are pleased to co-host this webinar with Bio Around the World, a platform that aims to streamline cross-border collaborations on global drug development by sharing real-life practice experience to achieve greater harmonization. Let's welcome Dr. Andres Cantobodis to our webinar interview. Andres, thank you for joining us today.
Hello, Echo, and thank you very much for organizing this webinar. Very interested to participate.
Thank you. We are so excited to learn more about your journey and your insights into the biopharmaceutical industry. So let's start. Could you please tell us about your background and how you get started Anaveon?
Gladly. So my background is I'm an immunologist. I'm a PhD immunologist. I started working as an assistant professor at the Georgia Institute of Technology in Atlanta, Georgia, doing mostly academic work. At some point, I decided that academics was interesting, but maybe having a practical work would be more interesting in the long run. I was very much interested in how our work in biology translates into medicine. And I had the opportunity to move to Switzerland at that time with Ciba-Geigy, which was a pharmaceutical and chemical company. I then moved into Novartis, which is in 2000, which was a full pharmaceutical company. My background is transplantation medicine and transplantation immunology. I have done work in autoimmunity, transplantation, and of course, the opposite of transplantation, which is immunosuppression, mainly immunosuppression, is the activation of the immune system against cancer. And that has been my major focus in my work the last 15 years.
Wow, what a kind of journey. So you have the extensive experience in drug development for immune-mediated diseases. What are some of the most promising modalities and approaches that you have seen in this space?
Yes, so in the space of tumor immunotherapy, of course, checkpoint inhibitors and new checkpoint inhibitors are being discovered and being used every month or almost every year, are very important. I have to say that we people that have followed the area have sort of realized that the first checkpoint inhibitors were a big quantum leap into efficacy. We haven't seen a second mode of action which has provided such big benefit yet, but I think now combinations are coming into sight. From my area, I think cytokines is a very important area where we understand some things, we don't understand everything, and there is one major problem with cytokines, and this is druggability. How do you make out of these small proteins druggable compounds or drugs which are useful, which need, as you know, stability, they need long half-life in the body, and they need to get into the tumor and provide immune activation into the tumor. In addition to cytokines, I think innate immunity is coming up. Quite interesting MOAs are coming up. And of course, right now, ADCs are something which is quite promising.
It's a very like exciting news for the patients. We have the more the treatment, innovative treatment, hopefully the cure the disease. So what do you think are some of the biggest challenges that still need to be addressed?
So a big challenge is how do you do combination therapy? We know that a tumor has a very, very differentiated genetic background. The tumor can evade the immune system. The tumor can mutate to resist different drugs. And of course, we all understand that it will be a combination therapy will be needed. Right now, you probably know better than I do that there are thousands of combination trials ongoing throughout the world. And to me, it's companies are now sort of working like throwing spaghetti on the wall to see what sticks, right? And do combinations without really having looked at the biology behind and make sure that the combination makes biological sense. So I think now, as we're getting more and more biomarkers, we understand how the tumor evolves. We understand what is important for us to attack the tumor. I think it will be time now to make combinations in a rational way and sort of have a better than chance to provide efficacy.
Thanks for you to sharing that's the opportunity and the challenges in this area. So you personally have been working on several drug candidates from the clinical trial to later stage commercialization. Can you tell us about some of the most exciting projects you have been working on?
Yeah, so we have been working on both antibodies, sort of anti-IL-1 antibodies, anti-IL-17 antibodies, which have been very successful in psoriasis. So I'm talking about my early experience, which was in autoimmune disease and transplantation. I developed an CD25 antibody for Novartis, which is still being used in transplantation. I'm very proud of that. And we have also been working on low molecular weight compounds for multiple sclerosis and also rheumatoid arthritis. Kinase inhibitors, these are very sort of diverse modes of action.
So you have also done the research on the immune cell traffic, as just you mentioned. So how do you see this field evolving in the coming years?
Yeah, for oncology specifically, there are important understandings which are coming up, including chemokines, which we know how cells go in and out of the tumor, activating cells to come in or go out of the tumor. And these are both good cells meaning what we call effector cells, which attack the tumor, or bad cells, which are cells like regulatory T cells, which protect the tumor. So we're starting to understand how cells get to the tumor, and we want to attract the good cells, and we want to take away from the tumor microenvironment the cells that protect the tumor.
So what do you think it has for the development of new treatments from this technology?
Yeah, so in terms of new treatments, like I said, at this point, we have a lot of cytokines which are coming up. The problem with them is that they are toxic if they are in a systemic environment. So you give them throughout the body. So there are technologies now which allow us to uncover the power of cytokines only at the tumor level. You may have heard them as corks or protected cytokines or releasing cytokine only at the tumor level. And so in general, specificity of treatment, specificity at the tumor level and not through the whole organism.
Thanks for sharing from all the advanced development in the science part. And also, I know you are not only just the expert on the science, but actually you have been so successfully raising the fund for Anaveon since the day one. So just in such a short time, you have been successfully raised over $100 million from the top tier investors. So what are the most important approaches you and your team made to achieve this success?
Yeah, so it's a typical Swiss system where we don't promise too much and we deliver more than we promise. So I think we built our investors by telling them an interesting story, which they believe. So the mechanism, the ideas that we have are original. They're better than what's around. And we then deliver more than we promise. So if we promise something, we try to not only deliver that, but much more than we have promised. Investors have been very good to us. We're very happy, and we're happy because these investors are not only looking for a return on the money, but they are company builders. They really want the company to succeed and sort of come up with a second and a third and a fourth drug. And this is exactly how we built the company right now. We have one drug in phase 2 right now. We have a second drug coming up into phase 1, and we have a few earlier drugs. So we want to be a pipeline company which will go from concept to proof of concept in man. We understand that we cannot deliver registration trials, trials that require 700 patients or 1,000 patients. These are too big for us. We intend to stay a small company which is produces a pipeline of new innovative drugs.
Thank you for that sharing. Like the market, especially the capital markets have been very tough for the biotech companies. So do you have any advice for the biotech executives who are looking for the fundraising?
Yeah, so it's very difficult right now where we are in the phase 1 and phase 2. But there's still a lot of money for early good ideas, and a lot of funds are raising a lot of money, and they have to put them somewhere. In our area, we see a change, and we see people starting to come back and say, what do you want to do and where do you want to go? But we are in an area right now where companies invest on data, they don't invest in promise. And until now, when there's a lot of money, any promising idea would get money. Right now, you have to produce good data. So my advice would be have a very good concept that you believe in, and also produce the data that make others believe in your project.
Is really like a data is like
data-driven.
Exactly, data means a lot. Is the key
exactly,
to make it a success. Thank you so much for sharing your personal journey and your insight about the advanced science development, and also your success of fundraising for Anaveon and take from the baby to today. So next, I would like to invite you to share more details of Anaveon, the pipeline, and the strategy.
Thank you very much. I'll have a few slides which will give a little bit of story of the company, but we'll focus mainly on the compounds that we're developing. Okay, so just to get started, Anaveon was founded in December 2018. We really started working in February 2019. And our original idea was to go into an area that we knew well about, and this is the IL-2 area, and to come up with a better idea than was previously used. I will go in, and then our development compound was this compound now called NV419, which we now have in phase 2. We sort of had an idea for the company not to expand too much, spend most of our money onto getting data and preclinical and clinical data, and not to have too many people. So we do try to keep a very lean company and not top heavy, which is actually spending most of their money into getting data. We were able to raise 35 million Swiss francs at that time on the promise of the compound. And then later in 2021, late 21, we raised another 110 million Swiss francs or dollars, it's pretty much the same, for the clinical development of this compound. In the meantime, we build up the teams, and we build up the preclinical team. One important thing I would like to mention from my experience is it's important to know what needs to be done in-house and what needs to be done outside. So for example, CMC, we don't do it in-house. We don't actually do GMP production, but we do most of our animal experiments, almost all our animal experiments, and most of our preclinical experiments in-house, because we believe that we have to have a certain quality and a certain reproducibility of data. Then in 2021, we got ANV419 into the clinic. We reached the maximum tolerated dose and the recommended phase 2 dose in 2022. And then we started the phase 2, so efficacy studies for these compounds. In the meantime, we have developed, as you can see, our original compound is mostly an IL-2. We call it systemic IL-2, whereas we developed what is now a next generation type of compound, which is a bispecific compound that has an IL-2 moiety, but it also has a targeting moiety, which is against anti-PD-1. The company has been very careful about getting very, very good managers. I will start with Gary Phillips, who is an MD. He has a lot of experience with multiple companies, including Big Pharma, Wyeth, Merck, Serono, Novartis, and he has led both commercial and other integration companies like Bausch & Lomb, and has, again, like I said, both commercial and strategic experience. He was at the World Economic Forum, where he was the head of the global health and healthcare initiatives, and he knows a lot of people throughout the business world in pharma. Our chief medical officer is Ed Gazal from the University of Zurich, an MD, a lot of experience in bringing oncology compounds into the clinic and to development. His last position was at Innovent. He was the head of USA at Innovent, and he moved back to Switzerland and joined us. So I have to say that these are very, very well experienced people, and it's a pleasure to be working with them and advancing the compounds with them. Christoph Huber is our Chief Scientific Officer, and he's also from the University of Zurich. He did his PhD at Scripps in La Jolla, California, and then he worked at Pfizer, and then he came back to Switzerland and worked at Roche. So you can see all these people have high pharma experience, and they're able to use the concepts that Big Pharma is using in the development of compounds. As I said, my background is mostly academia, early immunology, in biochemistry and molecular biology. Spent 5 years of my life as a professor, and then decided that I had to do something practical and not only teach. So our investors are, as you mentioned, fairly blue color, important investors who have been in the field for many years. Syncona and Forbion are big European investors. Cowen is a big US investor. And we have strategic investors like the Novartis Venture Fund and Pfizer, which are supporting us. We have built the company as a lean and efficient company. We'll not spend a lot of time on this slide, but we are based in Basel, Switzerland, and we are very lucky that there are a lot of pharma companies here, so we're able to get young people out of the university with some pharma experience who are enthusiastic, and they are interested in advancing and learning more about the business. And together with the senior management, which has a lot of experience, I think we have a very organization which is very dynamic and very, very innovative. The first compound we brought into the clinic was based or is based on IL-2, mainly because IL-2 is a well-tested mechanism. It is a well-known MOA. It had a lot of unknown parts of the mechanism, and when it was first developed in 1991, it was people didn't know much about the regulatory T cells or the T cells that are promoting cancer. And of course, IL-2 has both promoting cancer and anti-cancer activities. So although it had some efficacy, and which you can see here is 16% efficacy and 50% in renal carcinoma patients, which is now, of course, not dramatic, but it was very, very dramatic in 1991. The problem with IL-2 is that it has significant toxicity. It requires intensive care facility to administer the compound, and it has a low therapeutic index because it proliferates both the effector cells and the regulatory T cells. And of course, it has to be dosed 3 times a day because it's a small molecule, like we said, cytokines, very short half-life. So our idea was, could we do it, use IL-2, but do it in a way that it will be better? And to do that, we first have to direct the mechanism of IL-2 away from the regulatory T cells. On the left-hand side, you can see that IL-2 has a heterotrimeric receptor, alpha, beta, gamma, and which is present on so-called regulatory T cells. These are the cells that protect the tumor. And it also has a beta gamma receptor, which can signal only on CD8 and NK cells. So these are what we call effector cells, and these are the cells that have anti-tumor activity. So one molecule of IL-2 can have both pro-tumor activity and anti-tumor activity. So we thought, what if we could make an IL-2 which would not bind to the IL-2 receptor alpha, it would only bind to the beta gamma and will promote only effector cells. So this selectivity or specificity was something that would be important. Ideally, one would like to make the molecule also with a long half-life so that it doesn't have to be injected 3 times a day. To do that, we developed an antibody, and this is this blue structure right here, which binds. So this molecule, the purple molecule is IL-2. It's a small molecule. So we developed an antibody. It took a lot of time and searching for many antibodies, which binds in a specific part of IL-2, which is the one that the IL-2 receptor alpha binds. And we then made these complexes, but that was not enough. We had to sort of make the 2 molecules into a single molecule. So we were very happy when one of our designers was able to open up IL-2, insert 2 small peptides and connect it to the light chain of the antibody. And now we had a molecule which was a protein, and it had both the antibody, which protected IL-2 from the alpha receptor, but also was a single protein, which is important for the presentation of the drug as a useful compound in humans. So in the end, I will not go through the actual data. This is how our molecule looks like. It's a single polypeptide chain, light chain and heavy chain. And we have the crystal structure of the molecule. This is the IL-2. This is our antibody down the gray part. And the green circles are where they make contact. And as you can see, this is on the right-hand side is the crystal structure of the alpha, beta, gamma receptor of IL-2. And you can see that our molecule looks very much like the alpha receptor and IL-2 presented to the beta and gamma receptor. So in effect, we are producing something which is a single, like antibody-like molecule, which is able to act like the IL-2 receptor alpha and IL-2 together and activate the beta and the gamma receptors. So not only can we do that, but that also has an effect on cells. And what the effect here is that here you see the titration of proleukin. Yellow is proleukin, which is the native IL-2, human IL-2, versus our compound. And you can see that the affinity of activating CD8 cells is the same between proleukin and our compound. The other effector cells and NK cells, it's also, our compound is a little bit more potent than IL-2. But if you look at the tumor protective cells, which are called regulatory cells, our compound is 1,500 times worse than IL-2. So indeed, our compound is not as good in promoting regulatory cells or tumor promoting cells, but it's very good in promoting the activation of effector cells, CD8 and NK cells. This translates into efficacy into mouse models. We have not spent a lot of time on mouse models because we believe that humans are quite different from mice in this respect, but it's good to have some data. And you can see that in very sensitive models like the H22 liver cancer model, our compound is highly efficacious in protecting versus what the vehicle is doing. In less immunogenic tumors, we are doing okay. And in low immunogenic tumors, the compound has more difficulty. Our compound is the blue side. So we have pretty much fairly immunogenic tumors, we're very good. Not immunogenic tumors, we have more trouble in providing efficacy in the mouse. What we have shown is also that our compound, which talked about combinations, and what we've shown is that our compound can combine with anti-PD-1 and anti-CTLA-4 and provides increased efficacy in models when combined with anti-checkpoint inhibitors. Our compound is also able to provide ADCC support, and together with depleting antibodies like the anti-HER antibodies, we're able to get additional efficacy by combining our compound with our antibodies. So at least in preclinical models, it looks interesting, and we then decided to move into the phase 1. Just a little bit about the license, the IP, the intellectual property of our compound. We have 2 families of patents which have now been granted in most jurisdictions. We have also from China, we have a favorable reports. We keep the China alive in case we want to add more claims into the future. And we have an additional in the US. We have a third family, which now would extend the patentability of the molecule another 6 or 7 years from our original patents. The patents are relatively fresh. They're from 2016. A little bit about the CMC, or the production of the molecule. It's relatively easy right now. We do it at WuXi Biologics, who have been extremely successful with several projects with us. We have a very high producing cell line. We have purification that goes to 99.2% purity. And right now we have a solution, it looks like this, and we have -20 storage, but we're working on a lyophilization campaign, which should be finished by next year, which will be a 4-degree storage. So overall, just as a differentiation to some of our competitors who have chemical steps in the production of the antibodies, we are able to do everything on a cell line and fairly high production levels and also high purity. We started our phase 1 trial, and the goal of our phase 1 trial was to find the maximum tolerated dose and the recommended phase 2 dose. We started at 3 micrograms per kilogram and escalated all the way up to 364 micrograms per kilogram. I will not go through details. This was mostly a European study, and we have now reached the maximum tolerated dose, which is 364, and the recommended phase 2 dose is 243. We just announced, or we will announce that fairly soon, and we just came to this decision a couple of weeks ago. I want to show you one of the characteristics of IL-2 is that it promotes the expansion of effector cells. And I want to show you that indeed, this is from the phase 1 study in cancer patients. Our compound, you can see here, this is the low doses climbing up to the recommended phase 2 dose. And you can see that we have early proliferation of CD8 cells. And then it's well known that in the periphery, meaning in blood, proliferation is about 60 to 80% at maximum. So we've reached maximum proliferation early, and at the high dose, we still are safe, but we have reached the maximum proliferation required. The same thing with NK cells. You notice that the NK cells are more sensitive to our compound, and at lower doses, they respond well. We show that we don't have a lot of proliferation of regulatory T cells, which are the cells that we want to avoid. Something about the tolerability of ANV409 is very important to us because, as I told you, one of the important things that we want to differentiate to Proleukin is tolerability and safety. We have treated 40 patients, low-grade adverse events, mostly grade 1, grade 2. All of them reversible and manageable. effects were self-limiting, and they were responsive to supportive care therapy. The safety profile is mainly pyrexia, fever, some nausea, and then very few cases of liver enzyme elevations and lymphopenia. The lymphopenia, of course, is well known to be part of the IL-2 mechanism, and it's not long lasting. It's only 2 or 3 days. And mainly it's not a real lymphopenia. It's the cells going into the spleen and then coming back out again. Important, we have no discontinuation or due to drug-related adverse events. And we have reached, like I said, the maximum tolerated dose and recommended phase 2 dose. Important has come up from these studies that we have a fairly long half-life of the compound relative to IL-2, which has a half-life of 20 minutes. Here are the responses. Of course, I have to mention that the populations are patients who have been in multiple rounds of therapies and have failed multiple rounds of therapies, so one doesn't expect much. But you should notice we colored here the doses, and you should notice that as one is getting in more into the higher doses, we have more cases of stable disease and some partial responses, particularly in lung cancer. We've had one very interesting long-lasting partial response. This is the patient. She's a 63-year-old smoker, and she had, you can see on the left-hand side, the size of the tumor, pleural tumor at the start, and in a few months later, under treatment, this tumor almost disappeared. The formal reduction is about 60%, but you can see that the target lesions have both this one and this one have almost disappeared. So we're very happy that this patient had a long benefit from our compound in this case. So in general, what we have done is we have changed IL-2, which has short half-life. One needs to dose it at very high doses, reaching into high toxicity. And then, of course, it has to be done 3 times a day. Into NV409, which has a longer half-life, we can dose every 2 weeks. And due to the long half-life, we have good coverage of the CD8 and NK cell proliferation without reaching to too much toxicity. So we think we solved a lot of the problems of Proleukin. And in addition, if we compare how much IL-2 we are giving to the patient versus Proleukin, we can see that the recommended dose of Proleukin has a Cmax of about 28 nanomoles per milliliter. We have a Cmax of 31, fairly similar. But if you look, because of our half-life, the area under the curve exposure of the patients, Proleukin has about 300 nanomoles per hour times hours per milliliter, and we have almost 980 nanomoles. So we have about a good 3 times or almost 3.5 times more exposure to IL-2 than Proleukin. So I think this is important, and many people working in the area understand that the more IL-2 one provides, the better the chances for the patient to have efficacy. We're moving ahead with phase 2 studies, and we have a phase 2 study in melanoma and a phase 2 study in multiple myeloma, which should announce results early in 24. Everything is going according to plan in these. And these are also, apologies, these are also in clinicaltrials.gov if people want to have to see more details about these studies. So a little bit about competition. There are 2 ways. So a lot of other companies and investigators have realized that having selective IL-2 is a big step forward. And there are 2 major ways of making selectivity. One is by mutating the IL-2, and here you see the IL-2 to the interface of the IL-2 receptor alpha, and people make mutations. We call these mutains. And several companies have followed that, including Roche, Kymab, Neoleukin, Medicenna, and Synthekine. And we have on purpose avoided that because these mutains are highly immunogenic, and there's a very high risk of immunogenicity. And indeed, many of those molecules have failed because of immunogenicity. There is another way, which is what we followed, steric hindrance, meaning blocking the site of IL-2 binding by putting something bulky here. Initially, one of them was Nectar, which put non-specific pegs all around. This turned out to be that it was not very useful because this wasn't specific, the molecule wasn't specific, and the molecule was a prodrug, and they had quite a lot of issues achieving enough IL-2 or giving the patient enough IL-2. Synthorex, now Sanofi, put a selective peg onto a specific amino acid using technology developed at the University of California in San Diego. And this one also is not very selective because one peg is not sufficient to block the receptor alpha. Alkermes is fairly similar to ours. So they use the IL-2 receptor alpha to fuse to IL-2, different than we did. But in the end, the molecule is 45 kilodaltons. It has a low molecular weight, short half-life, and they have to give it daily for 5 days. I will not go into the Sodio, which is IL-15, but it's similar to, we put in the same category as IL-2, but again, it's a very small molecular weight compound, and then Sodio again has to give multiple times a week. Our compound uses steric hindrance, is high molecular weight, and has long half-life. If we look, and I will not go, this is a complicated slide, but I will not go through it. What I would like to show is relative to these competitors, we are providing more IL-2 than anybody else. And this is, we give you here the half-life of the different compounds. If you look at the amount of IL-2 that we are providing per cycle, it's more than anybody else. You should note that Nectar was not able to provide much IL-2. Notice it's 0.04 nanomoles per kilogram versus 2.74 nanomoles from us, and much more than all the other competitors. We believe that one of the reasons failure of Nectar is a low amount of IL-2 and the compound is non-selective. So what do we look forward with our molecule? I think we are looking for monotherapy right now in different indications. We expect that we will find possibly not first line, but I'm sure that third line or fourth line, we will find monotherapy. But of course, the real prize would be combination with other MOAs. So checkpoint inhibitors, enhancers of innate immunity, IgG1-driven ADCC, chemotherapy, and for different indications. We are working cell therapies. We are going to have clinical trials with TILs starting, and of course, CAR-T and NK cell engagers, and immunization protocols are just going to be important. We are working on all these areas. We haven't announced anything yet, but we will be looking for clinical collaborations in these areas. So overall, Anaveon has, looking forward, we have sufficient money to deliver an answer on compounds. We're looking for further investment to expand our indications and unlock the true potential of ANV419. We are in discussion with several pharma companies. We understand that we won't be able to do phase 3 trials ourselves. We want to stay a small company, and we are a small company. And we are now developing additional molecules further down against cytokines delivered directed to specific cells in the tumor, which I think is the next wave of innovation. Thank you very much.
Thank you. Thank you so much, Andres. I think your presentation actually is covered a lot of deep information, especially you spend a lot of time to compare it to other IL-2 developers. And I know this have been, your team have been making the meaningful progress. However, the IL-2 itself have been really challenging. So I'm not going to avoid such a challenging question. I will that put it up front. We know like recently have the heartbroken news from the Nokata and also Sanofi. You already mentioned in your presentation that there's a huge concerns. Actually, we can read so much news every day, even like in China, here around the world. And so IL-2 was well known, like you mentioned, the MOA and so many companies developed now. Actually, because of these two heartbroken news, and a lot of company actually facing like a bankruptcy or even like terminate the development. However, from your presentation, I see very clear the differentiation. And so I would invite you to talk more in the deep. How does your company L2 assets different from this the failed two assets? And what steps have you taken to address these concerns?
Okay, so just to your last of your question, we don't compare ourselves. It's it's very difficult to even make some of these compounds because the preparation is difficult. So we don't really want to compare them. And animal studies is not a very good comparison. And a lot of people have tried to do comparisons, and they put high doses. If you look at all these competitors, they've had efficacies at very, very high doses, which we know animals are toxic to animals, but mice are much less sensitive to IL-2 than humans are. So what we do, we have a more tolerable and safer compound. Of course, your next question will be why. So I don't have the answer to that, but it's high molecular weight, it's long half-life, and if in all the data that we have looked at, it acts on cells in vitro, in human cells, very much like IL-2 does. But in vivo, it's very tolerable. So in effect, we can provide a lot of IL-2. The other thing that differentiates us is that and other people have not done yet, including Nectar and BMS, was they went straight for combination therapy. We really believe that we have to find an indication and show that we have monotherapy efficacy, because you have to add 1 + 1 to make 2, right? You cannot add 0 + 1 to make 2. So in that sense, we believe we are looking for monotherapy efficacy at this point, and we have a safer compound which delivers more IL-2 to the patient. Maybe one of the points, if you've noticed that Sanofi, they have not stopped the program, but they said they want to go into intensification. I think that's a very interesting concept. We provide now, because we are tolerable, our compound is tolerable, we are going now to weekly schedule instead of every 2 weeks. Obviously, the patients don't like to come every week for this, but if we can show much better efficacy on a weekly schedule than the every 2-week schedule, I think people will take the therapy for the efficacy it provides. So we are intensifying, not only we're providing a lot of IL-2, but we're intensifying and providing it on a weekly basis for at least a month, and then we'll go into a bi-weekly basis. So I think, again, repeating myself, safety, tolerability, and high amount of IL-2 is what we differentiate from everybody else. And high selectivity. You may mention, I think, Synthekine, which I think has made the news in China, but Synthekine is actually totally synthetic protein, and they have high binding to the beta receptor. It's a technical argument, and I don't want to get into it today, but high affinity to the beta receptor is, I think, detrimental, and it's not good for the compound, as it also activates regulatory T cells. So one other important part of our molecule is it has natural affinity to the beta gamma and blocks alpha without changing anything else. So we do try, and also as a strategy, we do try to have minimal changes to the cytokine. And all we did was block alpha binding, but the rest of the molecule is natural IL-2.
So you just mentioned about like you are going to start the weekly schedule instead of 2 weeks. And so how the patients will benefit from that? Is it because you add a more frequency of the treatment?
Yes. So I showed you that we deliver about 900 micrograms in area under the curve. This will be double now. So patients will be able to get twice as much IL-2. There is sort of still in the area, is more better, and or is there a maximum? And I think we provide a lot on a every 2-week schedule. I hope that providing more on a weekly schedule is going to provide a better efficacy for the patients.
Okay. Oh, that really makes sense. So just like IO2, this MO itself, we know that when they were to get popular, we kind of like have the over 20, like in the preclinical trial, like also have the phase 1, have the 2 digital, like more than 10 in the phase 1. And so what do you see how this will be developed in the future? Because I think the company developed IO2 have been at a very high pressure, and so struggle, like investor may not suggest to continue even, right? So what do you see forecast this will be leaded to?
So the IL-2 area, you mean? So people are going into directed IL-2s like our ANV600 compound, and they're going to trying to intensify the non-directed IL-2s. We have another sort of advantage, and our molecule, as I showed you, promotes the expansion of natural killer cells, NK cells. Yes. We know that NK cells are not so important for melanoma, and it's a simplification. And I hate to say it because there may be immunologists in your audience, but we can see melanoma as a CD8 dependent disease, and we can see, let's say, multiple myeloma or hematologic malignancies as an NK dependent disease. So we are developing our compound also in multiple myeloma, where we think that the NK cells will play a big role there. So an additional difference between us and competitors is that we are expanding NK cells very, very significantly.
Okay, thanks for that sharing. So let's back to the Anaveon. So like, what's your next plan with move forward with the development of ANV 419, ANV 600?
Yeah, so ANV 419 will complete phase 2 studies sometime mid next year. We would like to partner it, possibly with a Chinese partner, maybe other partners in Europe or the US. We are pursuing multiple clinical collaborations with other companies. I think I showed you, immunization strategies, cell therapies. We are working on one, and we have a couple of others coming up. So we do want to find other partnerships that will show in the clinic how useful our compound is. And 600 is moving into phase 1 next year. Now, your question, I think, is a bit deeper, and it says, how are you placing the two compounds? So we are placing 419 more into immunogenic tumors like lung cancer, melanoma, head and neck, kidney cancer. And we will be placing 600. We haven't shown you data, but we have published some data. We're going to publish more data in a few months that poorly immunogenic tumors are responding very well to 600, because 600 is able to proliferate the so-called pre-exhausted CD8 cells in the tumor. So we expect that 600 will go into areas like breast cancer or other not so immunogenic cancers.
Okay, so that's really have the different direction for the.
It will be different direction.
Given the indication to leverage the advantage of the compound. So in terms of the 419, I understand your current clinical trials are in Europe, and so when will you expect
And the US, we have an
the
IND
US?
open. So yes, melanoma is recruiting very well in the US. So the multiple myeloma is only Europe, but melanoma has a lot of US sites open.
Okay, so when will you expect more the data will be available next?
Yeah.
The
Yes,
earliest?
so efficacy data will be available second quarter, first or second quarter next year, depending on how recruitment goes.
Okay, we are really looking forward to that. That
We
will
are very excited about it. Yes, yes.
and that will be really another like I think a change in the life of IL-2.
Yes, yes. I hope that this will be sort of like the next wave of IL-2. By the way, the climate has been changing. I don't know how it is in China, but if you go to the conferences here in the US and Europe, the climate is changing and people are starting to think of there will be new molecules coming up.
That's really true. Yeah, I even heard like we learned so much from the fail, and the drug develop a drug is not just the end game. Actually, is always learn from the fail, and then you redo. And so you take. That's why it's one successful drug. It takes 20 years, 100 million, more than $1 billion.
You're right. You're
Average,
right.
right?
You're right. You're right. So, and we have gone through that, right? So we were really scared that we're not the first, and then we learned from the mistakes of Nectar and BMS and Sanofi, and we sort of were able to change our strategy on the fly. So this is also the big advantage of being a small and flexible company, is that we can change our strategy very often to the environment.
Yeah. So I want to switch to gear here a little bit. Like you mentioned about it in your presentation, like comparing your IO2 assets to other IO therapeutics currently in the market or in the developer. So how will your treatment will be different from them? How do you compare it to the existing therapies?
Yeah. So I do not think that we're going to go first line to start with, right? So we're looking at tumors where so for example, lung cancer. I don't know what the treatment in China is, but in Europe, you have first checkpoint inhibitors, and then you have chemotherapy, and then the patient has nowhere to go, right? So as we are developing, and I cannot promise that this will be true, but we're thinking maybe go third line in lung cancer. So this will be something that, so people that have failed checkpoint inhibitors, have failed chemotherapy, can come to us. At the moment, they have nowhere to go. And it's about 50% of lung cancer patients are without choice on the third line. So we will start finding niches in third line indicate or whatever line where we are monotherapy, and at the same time expanding the combination therapies to try to move closer to first line.
Yeah. Wow. So you and your team is really under the high pressure. It's really life or death.
Well, I think this is everybody, and I think this is what life is in biotech. You have an idea, and you bring it as much forward as you can, right? And that's how the new innovations happen. It's I say it many times to people that ask me, are you sure it's going to work? I say, of course, I'm not sure, because it's science, it's nature, right? We don't know exactly how the disease works.
Yeah, that's really like a mystery. Even the doctor tell us the body is so mystery, we cannot tell why you feel
absolutely.
that.
Absolutely. It's actually a miracle that the body works. It's so complicated, right?
Yeah, so when we find one drug, that's why it works so much, and we wanted to make it available to as many patients as we
absolutely
could, right?
Absolutely.
So there's one of the key findings of the review paper. Actually, you have been spent a lot of time talking about you tried a different dose. So there's still not so much to learn about the optimal dosing, timing, and the combination of IL-2 therapy with other treatments.
Yes.
So like I can see you did a very different dose, very like in the very like not the bigger gap. So how do you design that approach?
Yeah. So that's a very good question. And people who are in the business, they know that the FDA is very interested. Until now, sorry, I'm changing subject. In oncology, people dosed at the highest tolerated dose. But the FDA is moving towards, it's called Project Optimus, trying to find the optimal dose. And so in our studies, we are doing 2 doses, a low dose and a high dose. And we have committed to the FDA to do even a 3rd dose to try to find an optimal dose. And there are some important parts in your question. For example, is it flat? So if you go to lower doses, do you get the same efficacy, but then the patient doesn't have so many side effects? Or is there maybe even a curve where there's an optimal dose and then maybe the efficacy goes down? From our animal experiments, we know that it's fairly flat in our case. You can give more, it doesn't get much better. You can give less and it's still good. So right now we have 2 doses in our phase 2 studies, and we will commit to explore more doses if we are successful.
Thanks for that. So in terms of combine the combo treatment, you also mentioned about like checkpoint and also the immunization. And so which one will be your first priority, you think will really make this combine different?
Yeah, so we have a study with a checkpoint inhibitor. It's not what most people use. It's a little bit different. And we have a study in cell therapy in TILs, which is starting in a few months. We are working on, with a company that is doing immunization. And we are working also with another cell therapy company where we are putting their cell therapy together with our compound. So we believe now it's time to go into the clinic, and we stopped doing animal experiments and we sort of try to find the combinations in the clinic.
So that's really that's exciting. It seems you really not slow down anything by the negative news, and it's still your team in the full speed for the trying the different ways to make this treatment for patients.
Thank you. We're very excited about it, and we think we like the mechanism, and we think it will be the compound that can deliver safe and enough IL-2 is going to win in the end.
Yeah. So you also mentioned about the collaboration, like you are open to the collaborator with other companies to license out or like a clinical trial collaboration. So what's your idea, the partners?
Yeah, so our ideal partners would be, first of all, very high quality companies. We don't want fast or companies that cut corners. We want a company that has the same ideas as we have. When you do something, you have to do it really well. We would like to have maybe expand our clinical trials to more indications. Right now we have, like I said, melanoma and multiple myeloma. We are very much interested in lung cancer. We're very much interested in additional indications. So a partner who can develop in other indications would be very, very interesting to us.
Thank you so much, Andres. I think Lisa, your presentation and the experience sharing and your insight of the Anaveon's, especially the leading assets development have been provided a lot of the valuable information to myself and the audience. And also, I think what are your really full speed work on developing this IL-2, I think is really bring the hope back.
Thank you. I hope we'll see it in a few months. Yes.
Okay. Thank you so much. And
thank you very much.
Yeah, I hope we will continue this conversation, and we were looking forward to your good news.
Excellent. Thank you. Take care, everyone.
Thank you. Thanks, everyone, for joining us today. And if you'd like to learn more about the development stage or the insights of Anaveon, you are welcome to reach out to Andres directly or reach out to my team. So we will continue the conversation. Thank you. See you next time. Bye.
Bye.